One goal of the MRSEC research is to understand the rules of assembly used by living systems in order to address engineering challenges. In biology, the best example of self-assembly is the formation of a virus, where hundreds of proteins assembled around a viral genome to form a protein shell called a capsid. Computational models, such as these coarse-grained simulations, play important roles in understanding the assembly pathways. The models allow us to see every detail of the intermediate structures formed during the assembly. In contrast, viral assembly is much more difficult to study experimentally due to the small sizes and fast kinetics of these assembly intermediates. Recently, there is an exciting experimental breakthrough which makes it possible to visualize the assembly of individual capsids around RNA molecules. However, the resolution is still not enough to distinguish the assembly intermediates with different structures. As a result, the experimental observation is the total amount of the capsid protein bind to the RNA, and the assembly trajectories are hidden underneath the noisy data. Now the challenge is, can we use computational models to infer the information about structures that we cannot get directly from these experiments? The answer is yes, using a technique called hidden Markov modeling. Imagine if you are graduate students and you want to know the weather outside, but your office does not have a window, so you cannot make direct observation. Instead, what you observe is that the professor next door is carrying an umbrella today. Luckily, you know the professor's habits of carrying an umbrella under different weather conditions. Now, if you have the knowledge about the local weather patterns in terms of how the weather today depends on the weather yesterday, then you should be able to get a statistical estimation of the weather even without direct observation. Similarly, in our capsid problem, all we need is a computational model to describe how assembly intermediates evolved over time. Then we are able to decode the hidden state evolution of capsid assembly from noisy data. The results shown here demonstrated that even with a very simple model, our method can statistically characterize the underlying self-assembly trajectories. We are excited about this inference approach, as it can potentially contribute to a more general framework for connecting biophysical models with material science experiments. Thanks for your attention, and if you are interested in more exciting research, be sure to visit the website of MRSEC.